My name is Barry Moore. I'm from the University of Utah. We've got a new group there called the USTAR Center for Genetic Discovery, and I think you've met a few of my new colleagues in that center recently. Gabor Marth, who's one of the instructors, and Aaron Quinlan, both instructors in the course, and some of the other people in their groups. So we're slowly working on just taking over the entire faculty of this course. If we can just convince Obi that the skiing is better in Salt Lake than it is in St. Louis, our conquest will be complete. So I'm going to talk to you today about a tool. I work primarily with Mark Yandel's group there in Utah, and I'm going to tell you about a project and a tool, really a framework called VAAST, stands for Variant Annotation, Analysis, and Search Tool. So we'll start off with just a little bit of a historical perspective. We'll keep that fairly brief, but billions of years of evolution have fine-tuned our DNA sequence. Alterations to that sequence we know can cause disease, and knowing which mutations are related to that disease can provide us clues for understanding those diseases and providing treatment. So we know all that, and for the last couple of weeks you guys have been working on the first part of this. What are the mutations? Next generation sequencing has been a boon for understanding that, and variant calling, while still challenging, is getting better. Today we're going to talk about approaches for deciding which mutations cause disease, variant and gene prioritization methods. So a really brief history of some of the milestones in medical genetics. When we're talking about variant prioritization and associating genotype with phenotype, we're talking about taking a look at genetic material in some form or another and trying to associate what we can see with a group of people who have a particular phenotype. So back in the late 50s, one of the first successes in this realm was understanding Down syndrome. So still an important field. Cytogenetics basically just involves coming up with some way to visualize the chromosomes, and we can see gross features like the chromosomes have arms and they have banding patterns, and in the case of Down syndrome, noticing that an extra chromosome 21 was associated with that disease. So still an important technique, but a fairly blunt hammer when you're talking about understanding genetic features. As the field of molecular biology improved, we were able to start identifying markers on the genome. So early on, simple things like restriction fragment length polymorphisms allowed us to visualize different places on the genome, even though we didn't know what the whole genome looked like. And following the genotype, phenotype as recombination broke the linkage down through families allowed you to narrow down portions of the genome that were associated with, in 1983, Huntington's disease, for example. This still would leave you with a fairly large window to look through, and so you had to use other techniques like positional cloning, a fairly brute force method to just figure out by linkage or other methods roughly where you are in the genome, and then just start sequencing one piece at a time and move along the genome until you can narrow down where you're at. And of course, this was all done back in the days of slab gels and radionucleotide sequencing, and so it was a very difficult and time-consuming, and it took years to sort out the locus for cystic fibrosis. Several large groups working very hard on that. Sometime in here, we got a copy of the human genome sequence, and that meant we could start choosing markers much more densely, kind of wherever we wanted. And so that was the era of the genome-wide association study, the array-based markers that allowed us to make a lot of discoveries in that domain, such as age-related macular degeneration. And then finally, next-generation sequencing comes along, and we have the ability to resequence entire human genomes. And a couple of the early efforts out of the Utah groups, Miller syndrome and Ogden syndrome, utilized those new technologies to understand causes of disease, or in the case of Ogden syndrome, to describe an entirely new disease based on next generation sequencing. So I'm sure you've probably seen this slide several times already this week. It's well circulated in talks and lectures, but I'm going to recycle it one more time. On the x-axis here, the time, and I'm a couple years behind. On the y-axis, a log-scale cost to sequence an entire genome. Of course, the human genome sequence was sort of released right around in here, and it had a budget originally of billions of dollars. They came in under budget, but it cost a lot. Shortly after the human genome sequence was released, Craig Venter quickly resequenced his genome for a cost of about $10 million on Sanger-based methods. Right about in here, so as the cost of sequencing continued to drop at the same rate of Moore's law, the law that describes the drop in the cost of compute power, we hit this inflection point where we're dropping off essentially logarithmically off a log-scaled graph, and that's the introduction of commercially available next-generation sequencing. Our local hero, James Watson, had his genome resequenced on some of those new technologies for an estimated cost of about $1 million. And today, we're hitting the cost of $1,000 for a genome. And really, any of us, if we really wanted to, could get our whole genome sequenced. In fact, maybe some of you have, or certainly an exome sequence for a price that we could afford, and certainly a price that's comparable to other medical techniques that our insurance pays for quite routinely. So it was right about this time here that we first released the first versions of VAAST, and trying to capitalize on this data to understand human disease utilizing this next generation sequencing technology. So in a cartoon form, this basic idea of genotype-phenotype association that we're trying to address, these little cartoon chromosomes will represent entire human genomes for different individuals, and if we sequence several of those individuals who have a particular disease or phenotype, we sequence several other individuals who are presumably healthy, at least don't share the same disease or phenotype that we have here. We sequence them and do the variant discovery methods that you guys have been working on. We are then going to go through and look for places where the variation is associated with the disease. So not so good, not so good, not so good. And we get here and we're like, okay, so maybe that looks like a good candidate. Of course, we don't expect to have, if these are unrelated individuals in particular, the kind of allelic homogeneity that would be the case if everything just all lined up. And so we can have rare alleles in different individuals contributing to the same phenotype. And that is more difficult to assay or to test for, but with presumably the entire genome or the entire exome sequence, we have all those rare variants, and so we can come up with strategies to do that. And one thing to do is to not just consider each variant individually, but to aggregate the burden of those variants, including the rare variants or the common ones, to the gene level. And we get a couple of nice features out of that. First of all, now we can consider all of these variants in these individuals together, even though they're not the same variant. And second of all, when we're doing lots of tests across the genome, instead of having to test every variant site and consider that an independent statistical test, which we then have to correct for that multiple testing problem at the end, we're now capped at testing for the number of genes that we're testing genome-wide, which is going to be fairly fixed and much smaller than the number of variants genome-wide. So that then still looks like it's maybe a good candidate for us, but we just need to be reminded that challenges still exist. So let's say this was an exome sequencing project where we had just sequenced the exome portions of these genomes, and we got these healthy individuals because our budget didn't allow us to sequence a cohort with this project on our own. For healthy genomes, we got this from a colleague who had done a different kind of capture, and that capture didn't capture so well in this region. If we aren't careful about how we process this data, that lack of information there could lead us to conclude that this is actually our candidate, when in fact what we really have here is just some missing data, and we didn't know that. Okay, so enough of that. We're going to move on to the tools that are part of the VAAST package. That, XQC, VVP, VAAST, and PVAAST are all different parts of that. I won't discuss every one of those in detail, but I will hit on the major tools. So just as a broad overview to the prioritization tools in the package, VAAST and its various forms are probabilistic tools for variant prioritization and disease gene discovery. They combine variant scoring with feature-based aggregative variant analysis, so they implement a burden test. Both allele and amino acid substitution frequencies are considered when scoring variants in genes. Scores are scaled by phylogenetic conservation, so we get some input from the experiment called evolution, not just from human populations. The tools implement numerous parameters and filters that allow us to kind of tweak the analysis in fairly fine-tuned ways. It uses a standardized set of ontology-based formats so that A, we can play nicely with other tools, hopefully, and B, using ontology-based formats, it allows us to take various sets of biomedical knowledge and compute on those in rational ways. It has a modular and flexible design. It tries to be an analysis ecosystem, and modular and flexible by that I mean that you can use different tools in different ways and combine them in different ways to come up with a lot of different analysis scenarios. So it's collaboratively developed by three groups: Mark Andel's group at the University of Utah, Chad Huff's group at MD Anderson in Houston, and Martin Rees with Omisha Incorporated in the Bay Area of California. I'll just let you see that there's a number of people who can contribute to the core development, and really all of these groups have been pivotal in putting together VAAST as it is today. So these tools kind of put together as a flow diagram. We want to start out with a VCF file that's generated by sequencing, and hopefully we have jointly genotyped all of the analysis genomes, all of the case genomes, together with a set of healthy control genomes. That joint genotyping really gives us a fairly standardized set of variant calls across the entire set and can help eliminate some of the technical biases that can plague these types of analyses. That is a functional annotation tool that comes with the package. It basically tells you where the SNVs and the indels affect features on the genome, like causing missense mutations and in-frame or frame shift deletions. VEP is another tool out of the ensemble group that will work as well. XQC is a QC tool that helps us manage some of those technical biases that can cause false positives in this type of analysis. And then the three tools, VAAST provides case control analyses, PVAAST provides pedigree-based analyses if we've sequenced a large family, and VVP allows us to just prioritize variants individually, and it also implements a burden test that allows us to very rapidly do a burden test on single genomes or small nuclear families where the one affected individual is involved. Okay, so the data coming in is basically the three main kinds of data types that we're used to in genomics: a FASTA file of the genome sequence, some GFF3 file of gene models, and then a VCF file of variants that are produced by tools like GATK's haplotype caller or Freebase. And the outputs would then be an annotated set of variants coming out of the VAT tool. The VAAST and VVP prioritized gene lists, and then there's a compact representation called a CDR file of the population variants that's used internally. It's really the same kind of information that's in the VCF file, it's just orders of magnitude smaller. So I'm going to just give a few slides to give a shout out to the sequence ontology. They manage the file formats for GFF3 and GVF, which is closely related to VCF, in standardized ways. They maintain an ontology of all the kinds of features, both gene model type features and variant type features. that we talk about in this domain, and they provide some software to help work with those tools. So one of the key things that they do to give a sense is that when we're talking about annotating the functional effect of a variant, we have 3 different elements that we are talking about. And not all tools that will do functional annotation kind of really break it down like this, but it is important that we realize that we have 3 different things going on here. First of all, we have a number of different types of variations that we can be talking about. So for example, a single nucleotide variant. Those features can overlap with a number of different types of elements on the genome, like genes and coding sequences and exons. And then those 2 things combined, we can predict different types of effects that SNV might have a missense variant on a particular CDS. But it could also have, because we have multiple CDSs for a given gene, it could be an intron variant in another transcript. So just to break that down a little bit in terms of how the sequence ontology manages this information, everything in the sequence ontology ultimately comes back to being a sequence feature. And we would intuitively know that, for example, a SNP, which is a polymorphic site in the genome, usually with some appreciable population frequency, is a type of nucleotide, single nucleotide variant. And we know that a single nucleotide variant, or a SNP, is a type of a substitution, and all the way back up. And the same sort of thing, that a CDS is a region of an mRNA, which is part of an mRNA, which is a type of a transcript, and all the way back up. So that's easy for us to think about because we've been trained with this sort of thing since probably elementary school or middle school. But the computer doesn't know this, and it's very difficult to tell the computer that kind of information. And these ontologies that not only constrain the words that we use to talk about these things, but also constrain the relationships between how these different features interact and are related to each other, is incredibly important for being able to compute on these kinds of data sets. So you can't really see it here, but a software package out of the sequence ontology, the genome annotation library, developed in the programming language Perl, allows you to write some fairly simple code that uses the ontology as its framework for understanding these sequence features and these sequence variants. And it isn't the kind of analysis tool that we're talking about today, but it can allow you to work with and prepare the data sets in an ontology-informed manner. Okay, so enough of that. I think you've probably already seen variant files and other types of files already this week. So we'll just briefly refresh our memory. The VCF file is where the variants are, some metadata at the top, tab delimited columns of information about each variant at the bottom. The initial data early on describes where the variant is, what it is, and a few bits of details about it. And then we have down at the end here this open-ended number of columns that describe for each individual in our data set how they are impacted, how they are affected with these alternate alleles that we saw earlier on. And the reason I wanted to just take a moment here is to just remind ourselves that when we call these 2 variants, when we genotype these 2 individuals together, we get information, for example, like this individual is homozygous reference here, but that same individual is missing data at this point. And if this were, for example, one or more of our affected individual, of our background individuals, and this was our affected individual, knowing that we're missing data here is extremely important to being able to prioritize the sites that are truly associated with the phenotype. Okay, GFF3, another file format, is you've probably met it or one of its closely related cousins during this course as well. It describes gene models, things like genes, mRNAs, and exons. And in particular, GFF3 gives you the kind of information in these last columns here to be able to say that this exon belongs to this mRNA and this mRNA, but not this mRNA. And so you can reconstruct an entire set of gene models for an entire genome using a file format like that. And then very briefly, I mentioned the CDR format, and it is less relevant to us as end users, but just to say that it contains that same kind of information that we saw in the VCF file about each variant. It's another, one row per variant, tab delimited flat file format. It captures annotation about those variants, and it's much smaller because instead of having a column for every single individual that might be in a very large data set, we just capture which genotypes have been seen in all those individuals, and then integer indexes tell us which individual is affected at that site. And so it allows you to cut the file size by an order of magnitude, usually at least. Okay, so the variant annotation tool, VAAT, adds functional annotations, for example, stop gain, of sequence alterations like the SNV caused a stop gain on a particular mRNA. It takes as input a FASTA file, a GFF3 file, and a VCF file, and it produces an annotated output file of VCF or CDR format for the tools downstream. So we'll be doing some of this in the lab this afternoon, but to just give you a general sense, -A, and we give it the genome, -F, and we give it the gene models. We give it the VCF file, and to standard out, it writes out the annotated VCF file. So I kind of already described this before, so I won't say it again, but the sequence variant is the effect. The sequence feature is the type of feature that is affected. And we also need to know which feature is affected, because a single variant can impact different transcripts in different ways, leading us to perhaps misprioritize the gene in general if we're not keeping track of which transcripts we're predicting these effects on. Okay, so VAAST, the original tool and the primary tool, scores, filters, and prioritizes features for their likelihood of being associated with a disease or a phenotype. VAAST, PVAST, and VVP each implement this same basic scoring algorithm, but address different workflows. And again, just a real quick example of a simple command line. There's a few required options. What scoring mode are we going to use? In this case, the default, the likelihood ratio test. Where is the output file going to go? And then again, we give it the set of genes, the background individuals as a CDR file, and the target or affected individuals as a CDR file. So let's talk a little bit more about that likelihood ratio test that I just mentioned as the default mode. Basically, with the likelihood ratio test, we're getting a ratio of two different likelihoods. In the numerator, we're asking what is the likelihood of the null model, and the null model here is that we have a set of healthy genomes and we have a set of affected disease genomes, and the null model is that there's really no difference between those two sets. And the alternate model in the denominator is that there is a difference, that for a particular allele or for a particular feature, a particular gene, there is a differential burden of variant scores in those two different groups. And then that ratio is just a log is taken of that, and that becomes the raw score for VAAST. So just expanding on that a little bit easier, I hate formulas, especially in talks, but I'm showing this one just to give you the sense that it's really just fairly simple information that's going into these likelihood calculations. These hat P's and 1 minus hat P's represent the minor allele frequency and the major allele frequency in the different populations. B and T stand for background and target, and we're talking about the allele counts, the major and minor allele counts in the background and the target. So up here in the numerator, we're considering the background and the target together for the likelihood that there is no difference between these two groups. And down here in the denominator, we have the backgrounds here and the targets here. We're considering them as two separate populations to determine the likelihood that they're separate. And again, we take the log and we can then sum those logs across the feature. So this is applied to every variant that's in the gene, for example. and then we sum that to get a score for that entire feature. Okay, so that's the probabilistic part of VAAST being a probabilistic tool, and then there are a number of filters and parameters that we can tweak and set to address additional information, biological information that we might know about this set of disease genomes or the genomes that we're looking at, and those can help us to add information that aren't strictly modeled in the likelihood ratio test itself. So dominant and recessive inheritance filters. In the case of even when we don't have family data where we're explicitly modeling the inheritance through the family, we can just assume that if we're expecting that we're looking at a dominant for a dominant allele, that we only need to score one allele per feature or one variant site per feature. In the case of recessive, we would want to be able to score two different alleles per feature, whether that's a homozygous or compound heterozygous. Complete penetrance, if this is an extremely rare disease where we think that there's no way that somebody who carries this allele that causes this disease could exist in our background population, then we can set a complete penetrance, and then we would not score any feature where anyone in the background shares the same genotype as one of the affected individuals, on the assumption that if anyone did, they would be obviously unhealthy. Rate and PAR, percent population attributable risk, will hold the minor allele frequency in the background or in the target at a given level. So in practice, you would apply that when you know something about the rate of the prevalence of the disease in the population. The minor allele frequency couldn't possibly be above, say, 0.1% or 0.5% or something like that. And so you can add that constraint here. And at the same time, if we had a large group of affected individuals who were unrelated, and we thought it's not possible based on what we know about this disease that every one of them is going to share exactly the same allele, we could put a maximum value on how much allele sharing we would expect. Those kinds of things. Mendelian inheritance, when we have small nuclear families, we can address inheritance in the family simply through filters by saying that the children need to have inherited their alleles from the parent, or in the case of, if we're thinking we're going after a de novo, they need to not have inherited the allele from the parent. So most of the tools that do the prioritization, actually all of them address those basic areas through filters or parameters. So in summary, VAAST is using the likelihood ratio test to ask the question, do aggregate allele and amino acid substitution frequencies, when scaled for phylogenetic conservation, differ between a background and a target set of genomes within a given variant or gene? The statistical significance of these scores are then estimated by a permutation test. Multiple test correction, as I mentioned before, then is relatively mild compared to scoring every variant site in the genome for your population. It's going to cap somewhere around 20,000 features if you're scoring genes in the human genome. And then the filters that I talked about can help you improve power by considering population or disease parameters that weren't explicitly in the model. So just a little bit about PVAAST, since it's a different kind of a tool, same basic scoring, but applies to a different type of problem. The P stands for pedigree, and so PVAAST incorporates pedigrees both large and small. You can combine multiple different pedigrees in the same analysis, as well as cohorts of unrelated but affected individuals. So if you can imagine a study where you might have 20 different trios, but you also have some sporadic cases as well, and you have reason to believe that those all should go together, you can do all of those in a single PVAAST run. PVAAST calculates a parametric LOD score that's designed specifically for next generation sequence data. And it also provides quite a number of fine-tuned parameters for things like penetrance, prevalence, disease prevalence, and inheritance. And those are explicitly modeled in the case of PVAAST because we have information about how the individuals are related to each other. So just very briefly a little bit about that LOD score. The PVAST LOD score differs a bit from a traditional 2-point parametric LOD score in that PVAST assumes that we're testing for the causative allele. So in the days of array-based GWAS analyses, you typically were expecting that you were looking at an allele that was somehow linked to the causative site. But with next generation sequencing, we typically think that we've sequenced the actual causative allele or alleles. And so the calculation is modified to take that into account. The alternate model states that the site that we're considering at this particular location, as well as a latent site somewhere else, are both causative and linked with some kind of differential probability. The Elston-Stewart algorithm is used to calculate the likelihoods for the LOD. And because there are a number of parameters to VAAST that can affect the ultimate LOD score, a grid search across a range of those parameters is done to maximize the LOD score for each site. Significance is calculated on that LOD score through a gene drop. So this is basically you take the pedigrees that you have and you sample without replacement new founders for that pedigree from all of the set of existing founders plus the control genomes plus any sporadic cases that you've included. And then you regenerate the pedigree each time in the gene drop permutation based on those new founders. You recalculate the composite likelihood ratio test and the LOD score for each of these simulations. And that permutation gives you an empirical p-value for that LOD score that's then directly integrated into the likelihood ratio test. VVP is the newest tool in the VAAST suite of prioritization tools. And VVP can place a prioritization score on any sequence variant anywhere in the genome, exons, introns, UTRs, regulatory, intergenic regions. VAAST can technically do that as well. VVP just takes a slightly different approach to that and can do that at the variant level. VAAST is not really designed to score individual variants. So VVP starts by simply scoring individual variants. It places 2 scores on each variant, a raw score and then a percentile score, which normalizes the score by each gene. So every gene is considered what's the range of VVP or VAAST scores that we see in this gene, and then where does this particular variant fall in that range of scores. That allows you to compare VVP scores from one gene to the next. Genotype data, if it's available, you can just score a file like dbSNP or ClinVar with no genotypes provided at all if you're doing that, if you want to do that. But if you have genotypes available, VVP will also calculate the impact of the gene function based on whether the site that you're scoring is heterozygote or homozygote. And it's just really fast. I mean, that's its biggest advantage over the traditional VAAST is that it can score an entire whole genome sequence in a matter of a few minutes on a single core on your laptop, whereas VAAST runs can take quite a bit longer than that, a full VAAST run. Okay, so that's a brief introduction to some of the tools. Now I'm going to talk a little bit about application to different types of disease gene discovery problems. So I'm going to start off with one that was actually the very first thing we tried to apply VAAST to, and it's 5 years ago now, so it's, I guess, ancient history in terms of this field, but still relevant for how VAAST can be applied today. We had a family coming into the hospitals in Utah. They'd been coming in for over 20 years. And in each generation, about half of the male offspring were dead by the age of a year. The family got to calling the boys their little old men because they had this progerid-like phenotype. It wasn't strictly progeria, but they did have an aged appearance, craniofacial anomalies, hypotonia, global developmental delays, and ultimately they would succumb to cardiac failure. So this family, the grandmother had first been bringing some of her children into the hospital. Then she had some daughters who had affected individuals. So we had 3 affected boys over 2 generations, as well as the grandmother, the proband, the mother, an aunt, and some unaffected boys as well. I mean, an uncle and an unaffected brother as well. We put this into VAAST, and this frame here at the bottom, which looks kind of empty, is actually a Manhattan plot with the log scaled p-value from the VAAST output on this axis and the chromosome on this axis, and we only got 3 candidate genes after applying the appropriate filter, and this was before PVAST, so it actually wasn't run through PVAST. It was just filter based on the inheritance through the family, and three candidate genes came up. The entire pipeline ran in a matter of minutes. NAA10 and acetyltransferase was confirmed by genotyping in the extended family and by functional analysis to be the causative allele. And when the paper was in review, one of the reviewers contacted us and said, "Hey, I've got the same family, and in our hospital here, we got to talk about this." And it turned out that that child had exactly the same allele, although the families were unrelated. So that's the kind of problems that we try to apply VAAST to, but that's one case. So to benchmark VAAST for the original publication, we randomly chose 100 known disease-causing alleles from OMIM. We inserted 1 or more randomly chosen published disease-causing variants for that gene into a personal genome. And then we assessed the ability of VAAST plus a number of other tools to recover those genes in this spike-in experiment. Repeat that 100 different times, 100 different genes to get some statistics on that. And this slide has the results of that. On the y-axis, we have the proportion of these spiked genes that each tool recovered. Each tool has 3 different columns. We've used 1 genome, 2 genomes, or 3 genomes to run the tool against. And VAAST and VAAST1 and VAAST2 both did very well, especially when you get up to at least a couple of genomes. The brownish color is the number of the percentage of the genes that were recovered in the top 10, the top 100, or the top 1,000. So the same kind of a thing shown with the new tool VVP. In this case, we're scoring ClinVar variants, and this is just variant scoring, not gene prioritization. But if we take all of the variants out of ClinVar, here I've selected in yellow CFTR, BRCA1, BRCA2, and then all ClinVar variants. And ClinVar describes whether the variant is considered to be pathogenic or benign. And if we run these back through VVP and score them, we can see, I'm going to start over here with BRCA1 and BRCA2, nice separation where the pathogenic variants are, the scores are clustered right up near the top. These are the normalized scores for VVP output. So on a scale of 0 to 100, where 100 is the most likely to be disease-causing and 0 is the least, the benign alleles are quite down here, and the same for BRCA2. We see an interesting case here with CFTR where the pathogenic alleles kind of have a bubble that is right down here around the middle. Even though we have these little white dots are our medians, we still have good separation between the pathogenic and the benign alleles. We have an awful lot of supposedly pathogenic alleles that don't look that seriously disease-causing. And probably there's a couple things going on here, but one thing is that CFTR has some very high allele frequency alleles in the human population. You know, one in probably 20 Caucasians carries a CFTR allele. So, you know, a few of us in this room may have CFTR alleles. So they're fairly high in the population, and because VAAST is using population allele frequencies, in these cases, it's thought that perhaps people who were heterozygote were less susceptible during the cholera epidemics of the Middle Ages and beyond in Europe, and so that we had some balancing selection at that time, and we're probably seeing some of that here. if we look at all of ClinVar together, we see a similar sort of thing, good separation overall, but this is more, these are called violin plots, but this is more like a lava lamp plot, where we see good separation between the medians of the scores, but some alleles that are considered pathogenic are still scoring low, and some that are considered benign are still scoring high. And the same kinds of things can be going on there, but this does include everything in ClinVar, which includes things like drug resistance, which we don't expect to be necessarily very rare alleles, and things that are in ClinVar that are described as unknown. So we're reflecting some of the ambiguity that's in ClinVar as well as that.
Is this blind to the incidence of, because your point about cystic fibrosis is well taken, but that should also be taken into account because cystic fibrosis is not a rare disease compared to a lot of these other diseases. So does this not, at this point, you're making the point that pathogenic, known pathogenic CFTR alleles are not uncommon in the population, and perhaps that's why some of those known pathogenic variants are getting lower scores.
Right, and this is, go ahead, sorry.
But shouldn't we take into account the incidence of the disease and
Yeah,
score them
right. And so this is just VVP running on score the variants, and don't give it any other information. In fact, it's running off the ClinVar VCF file, so it doesn't even know genotypes. So for example, delta F508 is very common in the population, and actually looked, but yeah, actually we did look. Delta F508 scores around 60, right around here. But that's scoring it as a heterozygote, and it's fairly common in the population as a heterozygote. If you score it as a heterozygote, if you score it as a homozygote, so if you tell VVP even the simple information about genotype, it scores 100. So it goes right up. So VVP, when it sees it as a heterozygote, because I'm scoring all of ClinVar and you haven't told me genotypes, by default I'm just going to score everything as heterozygote, then you're heavily affected by the allele frequency. But if you give it the genotype information, then all of a sudden it's the genotype frequency, and it scores more appropriately. Okay, so VAAST can also be used for large case control studies. We haven't had a lot of really large case control studies to publish these results on, and so this power comparison is done on a published NOD2 data set where the group had gone back and resequenced several hundred individuals just for the NOD2 locus, power on the y-axis, and the number of individuals in the resampling on the x-axis. VAAST and VAAST2 are here along the top, and we can see that even after, with alleles and a causative locus that's considered to be fairly common disease-like in the population, when we're out to 100, 150 individuals, VAAST is starting to gain really good power, whereas some of the other tools were not doing quite so well and needed a larger number of genomes sampled to produce the same kind of power. So PVAST, I mentioned this a little bit before, but it extends VAAST to incorporate pedigree data. I already described the design of the LOD score, and that the LOD score, once calculated at each locus, is then incorporated directly into the composite likelihood ratio test. And in large-scale simulation studies and reanalysis of known disease pedigrees, PVAST did significantly well, certainly outperforming VAAST, and I'll show you a slide on that. So here's a large pedigree. This actually wraps around, so this is one half of the pedigree, and this is the other half. It's a large Utah family that suffered from early onset atrial fibrillation was the target phenotype, but there was also some long QT syndrome and other phenotypes, cardiac phenotypes in the family as well. It was originally started out as two different studies, this family and this family, because the two families didn't know they were related. But once the study got underway and we plugged these individuals into the Utah population database, we were able to realize that there was a founder several generations back, and so the project was combined. PVAST was able to analyze this data set and come up with a causative allele. And I just want to show you an example. This is another Manhattan plot, and this is that family with all of the affected individuals just run through VAAST by itself. So all of the dots in this case, we have the the log scale p-value on the y-axis and the chromosome location along the x-axis for the entire genome. Each dot represents an individual gene in the genome. And we have some good separation of a number of alleles up here at the top, but they're all plateaued. And I think this permutation ran for a week or so, and we're still getting a lot of genes that are just right up there in the plateau. And the answer is in there somewhere, and it's actually not too bad. You know, we could start going through that list. But the reason there's so many genes that have that same score at the top is because we're running this through just VAAST that knows nothing about the relatedness of these individuals. And related individuals share a lot of alleles in common simply because they're related. And so that we're seeing that effect right here. When you run this same data set through PVAST that knows about the relatedness between those individuals and can calculate linkage, we get the top hit is the gene that they went on to show was causative. And so that's the power of pedigrees and the power of adding PVAST to the mix. Yeah.
How many, is there a minimum number of family members that PVAST needs in addition to the program?
No, you could run them on a trio. It's, you know, in the case of a trio, your LOD score is really binary. I mean, it's yes or no. Does it fit or does it not? So having more informative meioses certainly increases the power. And that's why we've implemented in VVP VAAST originally, but now in VVP trios and quartets, and because you know you can really do that by filtering.
Well, it looks like in this picture though there are some other variants that are probably not pathogenic that are getting upcalled along for the ride because they're linked.
Because of that linkage, yeah. So that's actually this particular one shows quite nicely that linkage comet trail coming off that causative allele. And probably not the primary concern of most people here, but maybe someone is. You can use VAAST. We tried very hard to design from the very beginning that this not be a human-centric tool. You can use it in other organisms, and people in our group and in other groups as well have done that. So here's Mike Shapiro in the biology department at the University of Utah studies phenotypes in domestic pigeons. And all of these pigeons share a phenotype that's called a head crest, whether it's a little calic or a hood where the feathers just entirely cover the pigeon's face. These are domestic pigeons that are bred by pigeon fanciers to look this way, but it turns out to be a fabulous resource. And in fact, Darwin himself was very enamored with this sort of studying selection in breeding populations of pigeons and other organisms. And so there's B and C here shows an F statistic analysis looking for fixation along the genome, and it identifies a region here. VAAST run on that, identifies a location right here. Both of these have dotted red lines for genome-wide significance. This little area down here is a zoom in on that fixation statistic, and we can see that it kind of gives us a couple of broad humps here in this general region, of which a number of genes might be involved. But VAAST just cuts to the chase and says it's this variant in this gene. And Mike's group went on to show that, in fact, that was true. So that's all great. And I've shown you some nice Manhattan plots that convince you that VAAST is doing a really good job. But next generation sequence is problematic and noisy. Variant calling is difficult. This is a QQ plot that shows if we just do a VAAST run on a data set of next generation sequence data, a QQ plot basically compares the distribution of the p-values to the normal distribution. So here's the observed p-values in log scale on the y-axis and the expected p-value on this. And so if p-values were completely normally distributed as they ought to be, we'd be right along this line here with some kind of a confidence interval around that. And we have a real serious problem of type 1 inflation here because our observed p-values on the log scale are higher than we would be expected by chance. So in real world terminology, this translates into a whole bunch of false positives that we now have to dig through to sort that out. So even though joint genotyping can reduce those biases, type 1 inflation can still be a real problem. And Chad Huff's group, Yao Yu and Chad Huff's group, has developed a new next generation sequencing QC toolkit that helps mitigate the effects of these systematic NGS technical biases. I won't go through in a lot of detail. I'm running a little behind the XQC package because it's A, just out, B, we haven't even released it yet, B, we're not going to work with it in the lab today, but I'll just roughly describe some of the things it does. If we take jointly called variants in a VCF file from hopefully both our targets and our control genomes that we're going to ultimately use, called on GATK or Freebase or whatever, we pass them through the pipeline. Won't go through every box, but basically at the sample level it has tools in there that will help detect gender misidentification. It will analyze missing genotype rate, so how many individuals in our groups are no-called at that particular site. Some general QC metrics like threshold for alternate allele, supporting reads, Hardy-Weinberg calculations, and then cross-platform QC metrics. So if we have different platforms, sequencing platforms in our data set, that sort of thing. And then it does a two-stage PCA, principal component analysis, to help determine what are the primary variables, the primary covariant components in the data set, and it'll build a covariance matrix file for you as well as a variant mask data set. In some cases, you might exclude some low-quality samples and fix some gender misidentification and run it back through the pipeline. But ultimately, after the PCAs, you get a covariance matrix file, a masking list for sites that are just too problematic to consider scoring and so forth. And then you can put those outputs, and they'll come out in VCF or CDR format. You can put those into VAAST, PVAST, or even other tools like SCADO that are helping you prioritize this space. And looking at QQ plots on data before, where we had quite a lot of type 1 inflation, and data after, it's still next generation sequencing data, and so we can see some consistent inflation, type 1 error rates, but they're much, much better. And just to show that, you know, it doesn't just work, the output work with VAAST tools, SCADO before XQC analysis and then SCADO afterwards, and you have much better performing p-values. Okay, so then in the last few minutes here, I'm going to just talk briefly about some related tools. The first one is another project out of Mark Andel's group. The tool is called Fever. It's a phenotype, ontological phenotype re-ranking tool is kind of the root of that acronym. And you take information on the phenotype out of a human phenotype ontology, and you can include the gene ontology here as well. And then you take a ranked set of genes from a tool like VAAST, but this could be a ranked set of genes from any kind of a tool, and you put that phenotypic information and that ranked set of genes into Fever, and it will re-rank the genes based on the likelihood that the genes in that ranked list are associated with the given phenotype. So I'll give an example here really quick to just show you how that's applicable, and then we talk very briefly about what it's doing. So this is a single exome that has been run through VAAST, and this is a difficult case because we're asking a tool to go through just a single exome and pick out what's the causative allele amidst the genome-wide background of noise that we expect in next-generation sequencing data. So two things to notice here. This is very stratified. These are, again, this is a Manhattan plot, and so it's log scale p-value on the y-axis here. We see a lot of just flanks of stratification in here because the p-value is not very well resolved, because with just a single exome, the p-value is calculated by a permutation test, and you know, we only have background data sets of, say, 1,000 genomes, like we can get from the 1,000 Genomes Project, or maybe some of us have data sets in-house that large now, but in general, we don't have tens and tens of thousands of control genomes that would be needed to permute enough times to get a genome-wide significant p-value. And so we have a real plateau of potentially causative alleles here at the top, and the answer is in there, but we have to go through that entire list of genes to try to figure out the answer. But we know something else about this. In most cases, we know the phenotype that we're chasing, and we haven't considered that yet. And so we plug a phenotype in with fever, as well as that rank list from VAAST, and it downweights genes that don't appear to be affected or associated with that phenotype, and it upweights genes that do appear like they could be associated with that phenotype. And so a new Manhattan plot shows that we now get nice clear resolution on the ABCB11 allele, which in this case the phenotype was intrahepatic cholestasis.
So can you define your own phenotypes to use this software? So if you took, all your gene stocks and rank ordered them or give them a score about how, curly they are, any random phenotype you want to define, can you then go back and use this to try to correlate that with, say, exome capture or something? Or is it?
You'd need to have those phenotypes somehow structured in an ontology to be able to use this tool. So basically the way it works is you take the genes in the list that come in from the prioritization tool like VAAST, and most of these ontologies will have some kind of gene to ontology term association file. And so for example, in the human phenotype ontology, you would take the set of genes that came in as scored by VAAST, and the nodes that were associated with those genes would get that score. And then that score propagates throughout the ontology, so that even if you haven't hit a gene that is known to be associated with a particular disease, the scores propagate through the ontology away from that node. So all of these different parts of the ontology are then, the nodes are kind of getting lit up by scores that are propagating from genes that were prioritized. And when those nodes cross paths, that score can amplify, so that as you move up the ontology, you can aggregate those scores at higher levels. And then at the same time, because the phenotype ontologies or the disease ontologies aren't, there may only be a few thousand genes in those phenotype ontologies that have those kinds of mappings. The scores are propagated through the gene ontology as well, and then the connection between those ontologies, the scores pass in between. So you get basically, at the end of the day, the effect that you get is all of the genes that are in your prioritized candidate list propagate through a disease ontology or any phenotype ontology. It doesn't have to be disease like you're describing, and propagate through something like the gene ontology, where you're talking about biochemically similar or functionally similar genes. And then everything is re-ranked based on the ultimate score that the genes associated with those nodes get. Makes sense? So you could do that in non-human stuff or in non-disease phenotype areas, as long as there was some kind of an ontology, structured ontology to compute over. Okay, so then, yep.
Yeah, so this gene, this is an interesting one for me. There are actually 3 different clinical diseases that are of limitations. Gene 1 is kind of an older one than intrahepatic cholestasis of pregnancy, and then there's PFIC, which is a progressive familial liver disease in a child, and also BRIC, which is a benign variant. And all 3 of those are kind of different mutations. So based on this, is there anything that you can tell us in regards to which of those 3, or now that we have the gene, do we just have to do a different kind of analysis to try to figure out? Because certainly you get clinically, but sometimes there's some clinical overlap.
Yeah.
Benign form, for example, and the cholestasis of pregnancy.
Yeah. So in this particular case, the phenotype was described by our colleague Steve Guthrie in the School of Medicine, and he gave it the diagnosis, the child, the diagnosis of intrahepatic cholestasis. So that's what he was going after. This tool is not really going to help you tease apart the those differences in which phenotype you're looking at, because that's the input to this tool. So you come to it with some kind of a preformed opinion about what the phenotype that you're looking at is. I mean, you could probably have put in, well, you certainly could have put in any of those phenotypes, and this gene would have, been weighted up, because I think all of those would be associated with this node in the ontology. So there's a little bit of maybe a little bit of an art to figuring out what phenotypes to put in. In general, the more specific phenotypes you can put in, probably better. But sometimes people want to put in just every phenotype term that they can think of, and that can have the effect of spreading, lighting up so many parts of the ontology that you start to lose resolution. So I think in general, the way we do it in practice is we'll go to the human phenotype ontology, for example, through the phenomizer interface, if you're familiar with that. And you can just start typing in clinical features, and it will suggest terms in the ontology that those features have been associated with. We use that as a starting point, and often it's sitting down with the clinician and saying, okay, let's just look at this together. What are terms you would describe this patient as having to begin with, and let's choose a set of terms that will weight all of the relevant parts of the ontology, but not scattershot it so much that we haven't really given ourselves any resolving power at all. Okay, so just in closing then, I will describe a tool out of the group, Martin Reese's group at Omisha, a company, a biotech startup in the Bay Area. So they've been active in VAAST development from the beginning, and their target is really the clinical domain. So medical geneticists at clinical diagnostic labs, or perhaps even physicians who are genetically aware and have genome sequence in a hospital. And you can't see this well, but this is a data set where a number of different trios have been uploaded, and we can go through and do things like in this drop-down box over here, you can see that you can launch an app. I can select one of these genomes, and I can come over here and I can trigger a solo or a trio or a quad VAAST analysis. I can trigger a fever analysis. Once I have a VAAST run, I can choose that VAAST run and add the phenotypes, and it makes it all, there's a lot of people who could benefit from tools like these who are never going to touch the command line, and we develop tools for the command line. These guys try to wrap those tools up, make them robust and easy to use for a clinical domain. So just a quick example, we'd go in, for a quad. It's really nice and easy to use. You just click on the father and you get a pop-up list of the genomes that are part of that project, the mother, the proband, an unaffected sibling, and you get to select the genomes that fit those boxes, and it checks to make sure that you didn't put a female in for the father and that sort of thing. And then you check over here the background file that you want to use, you click go and you come back. We've just re-implemented a new version in here based off of VVP for these. You click on it and basically, as you're getting back to the next screen, the run is finishing, and within a minute or so, you can see the output. And the output then, I don't know if you guys can really see this that well because of the colors and the resolution, but the output is in this basic web format where you have each allele with information about where it is and what the effects are, and this is missense. And in this case, this is taken from a trio, so we can see the zygosity of the child and each of the parents. We can see that these alleles were in HGMD and ClinVar and weighted with scores by other tools like CAD and SIFT and mutation taster, and then Omisha aggregates that into one score. And so this was a test case that I'm showing you here where the spike in was in CFTR, and it pops up to the top of the list. It also runs them automatically in recessive, because this is was a trio, it ran it automatically in recessive, X-linked, and de novo, and we can just select any one of those and view this. So this makes a really nice interface for users who would never be willing to get down and run things on the command line and look at text files in, that sort of thing. And then also, I was just going to show that the link outs that are provided on that screen will take you directly, for example, to the UCSC genome browser, or give you a little pop-up window of that particular gene, if this was a new gene to you and you didn't really know what it was, with NLP phenotype mapping, giving you terms and that were found in the literature, and PubMed IDs to link out to find out. So it can really, it's both. I like to use it now too. It's gotten good enough now that it's, you know, for a simple case, I'd rather just do it there than do it on the command line myself, because it's less copying and pasting to look at things in the genome browsers and to look up disease or gene names that I don't know about. So in summary, VAAST is a probabilistic disease gene discovery framework. It's feature-based, burden tested, integrating both allele and amino acid substitution frequencies, conservation, and pedigrees. It's got lots of parameters and give you fine-tuned control over information that you know that may not be modeled directly. XQC provides a robust QC covariation framework, single exomes, complex pedigrees, case control analyses, and everything in between. Fever integrates, actually that's a typo there, I've copied and pasted something there. Fever integrates phenotypic information, and OPAL supports clinical applications and integrates clinical diagnosis to make it easy to use for people who are more comfortable in a windowing web browser environment. And VAAST is attempting to be a complete analysis ecosystem for these data sets. It's under active development by 3 different groups, and there's support through a active mailing list and so forth. So, all right, and then I'll just finally close by saying that you can get at all of this by going to the Yandell lab. You could get at it through the Huff Labs website at Houston as well. They have a similar setup, software page. You go to that software page and there's links out to the code and the documentation and the mailing list and all of that sort of thing. Just to acknowledge there's a whole lot of people that are involved in the development of VAAST. They come from Chad's group, Mark's group, and Martin's group. And then I gave a shout out during the talk to the Sequence Ontology, who I think does really good work and whose work is very important to being able to compute on these kinds of data sets. This is getting to be a year or 2 old now, but there's groups from all over the world who have downloaded the tool and are using it. So we'd love to add a dot to your institution on that map for the next time. All right, thanks a lot. I'll close up. It's gone over a bit, sorry.